Good morning and welcome to the Elixir Friday Coffee, where we're trying to present the portfolio of services and tools we are offering to users as an infrastructure. Today is a bit different because today's topic is about an emerging field where bioinformatics and life science as a discipline are merging or probably having something in common. Lucie Houdová from University of West Bohemia, who are also trying to pursuing the field of precision medicine in the Pilsen, will try to introduce you where is the interface between the bioinformatics and the data and the precision medicine in terms of the healthcare. We suppose that this is just an introduction, and later, probably in the second half of the year, Lucia and her colleague will be presenting specific tools which they are actually already using or supporting the colleagues from the medicinal faculty in Pilsen. So Lucia, please, floor is yours, and we are looking forward for your talk.
So thank you for introduction. Good morning, everybody. So how we heard from Jiří, I will tell you something about Elixir check possibilities for precision medicine and our future plans. So at the beginning, I would like to introduce you the field of precision medicine and the interface between precision medicine and bioinformatics. And then I would like to tell something about provision of effective bioinformatics support focused on precision medicine. So it means during the research project or during the clinical study development, then during data collection and its generation, during data analysis, and at the end about its application in clinical practice. So what's precision medicine? Precision medicine is approach to optimize medical treatment based on patients' genetic, environmental, and lifestyle aspects. And what is necessary to mention that this has a strong impact on knowledge-generating healthcare and research participants. So it means when I will talk about the data, I will be talk primarily about human data. And because bioinformatics is the field that is focused on storing, retrieving, organizing, processing, specifically omics data, it will be more focused on genomics data, transcriptomics, or proteomics data. During my presentation, I would like to show you that clinical research and clinical practice is fundamentally collaborative and multidisciplinary effort of clinicians, biologists, and bioinformaticians. So this presentation is primarily intended for clinicians, for medicine specialists, and biologists to show them how can we want to help from the point of view of bioinformatics. So for my presentation, I decided to use as fundamental material the paper 10 Simple Rules for Providing Effective Bioinformatics Research Support. Because I think this paper consider all the point of views of bioinformatic help for clinical support in a very nice way. So these 10 simple rules, we can divide it into 3 parts. The first is project development, the second is data collection and generation, and the third one is data analysis part. In the first part, it concludes 3 rules. The first one is collaboratively designed experiment. So I would like to show you that during the clinical research and then it's the results application into clinical practice, it's a big effort to collaborate together, the clinicians and bioinformaticians from the beginning, from the design of experiments. The second rule is about manage scope and expectation, and the third one is about defining and ensuring data management. So just a brief reminder how the research life cycle looks like. The first step is always a question or some hypothesis definition. The second one is setting priorities of measurement of data collection. The third one is data collection itself. It's followed by data analysis, and the results of data analysis has to be interpreted from the clinical point of view. And of course, these results has to be disseminated. It doesn't mean only published in some papers, but in this case, it means its application to clinical practice. So, I want to show you that in each part of this life cycle, a bioinformatician and ELIXIR as infrastructure itself can help somehow, and we want to focus there. So the first rule is about collaboratively designed experiment. Every well-designed experiment has to start with well-defined hypothesis, and it has to cover also sample strategies, data handling, and reporting. In the precision medicine, the critical role is on the side of appropriate sample size. It's not just about the money, but it also can help reduce the types and sources of variability of data, can increase the generalizability of the experiment, and of course, it make experiment replicable and reusable, which is one thing we all needed. During collecting the human data, there can be problem with huge volume of data of huge size, because, for example, if you work with high throughput single cell RNA-seq methods during the experiment. So it's significant to consider how big the sample size the cohort should be. It has also significance and impact to data analysis, and of course, it's always about money efficiency. The second rule I just mentioned briefly. The second rule is about communication. So it considers that bioinformaticians and clinicians should work together, and there is a necessity of have the same language to understand each other, because at the beginning of preparing some study, it's good to have analytical study plan which covers research topics, but not just this, also some potential limitation, including technology, resources, and analysis. So it means when we work together, there can be some benefits for all of us, and especially for clinicians. And if the analytical study plan is well considered, it also has impact to management during conducting the experiments. The third rule is about defining and ensuring data management. And this is the field I think we can help currently some way and can help more in the future as bioinformaticians. Because as you see on the picture on the left, on the A part, you see the research life cycle as shown before. The B part shows the life cycle from the point of view of data. So you can see that there's more steps from the data point of view during research life cycle, and this all has to be covered. So it's good to have a data management plan, which should contain how data should be organized during experiment and its collection, how quality control is ensured, how data are stored. It should cover handling of data during the experiments, but also from the long-term point of view. And in the case of precision medicine and clinical studies, it also has to cover legal, ethical, and also funders requirements. the data management plan should cover a type of data which will be used, standards and ontologies, and it has to promote findable, accessible, interoperable, and reusable research. So it should focus to have everything prepared to verification process. And this plan should be prepared together in cooperation of clinicians and bioinformaticians, but also can be used existing bioinformatics tool. And one of them is, for example, data stewardship wizard, which can ELIXIR offer currently, and you will hear about this tool, I think, next Friday more. So the next part is about data collection and generation. This covers managing of traceability of data, metadata determination, and coordination of data and its security. Overall, this covers data interoperability, its accessibility and understandability for humans and also machines, its reliability, usability. It can ensure identification of sources and prevention of productions of erroneous data due to covering traceability, validation of collected data, and also data storing, data security, including data transmission, and of course, if it's necessary, access restriction. For these is a advantage of having data management plan, because it should cover all these parts of data processing. And of course, in this case, it's needed effective bioinformatic support. And it means if you have sustainable infrastructure which cover this, it's a big help for clinicians. So, and ELIXIR is such as sustainable infrastructure and has tools and services for covering mention. Just one thing I would like to mention more detail is rule number 5, which is about determining metadata, because metadata are essential for verification process. And how you see before, on the data process cycle, one of step is finding accessible data to your experiment. So if you, as clinician, make some experiment, it's good to public your data, but you have to describe them in a correct way to be maximally exploited in the way which is somehow standardized. It also can be used as a base for develop and implementation of analysis and visualization tools, which can be also then used in your in practice. How the metadata should look like, it depends often on the dissemination process, because some journals have some requirements, there are defined standards, some defined structures and schemas, and this is the part where bioinformatician could help. What kind of data has included in metadata? It depends on experiment, it's experiment specific. So if there are a few methods, for example, MIME, MindSecure, or MyAPI, it depends if you work with DNA, next-gen seq data, or proteomics data, and so. But you always need effective system to ensure comprehensive metadata reporting. And so there could, in this part, bioinformaticians can help. The last part is about data analysis, which covers 4 rules. These rules are basically about tools and about possibilities for data analysis, for data quality control, for covering the changes of workflow of experiments and repurposing the collected data. I would like to mention that part of analytical study plan should be quality metrics definitions because there has to be quality control through the project life cycle, the whole project life cycle. So it means on the side of wet lab, also inside of silico lab during the data processing, and the procedures has to be on the level of sample as itself, but also on cohort level during the clinical study. And the choice of computational tools of data analysis is very important, and in the field of precision medicine, the tools are very specific. So no tools is expected to be the best for our situation, and it's crucial to employ appropriate bioinformatics method and tools, and if it's necessary to develop a new one for a specific experiment. And what to do if something changes? There's always necessary somehow to ensure that the handling data during the changes. And if it is possible, you can also repurpose your data even if they do not be as suggested on before on beginning of experiment. And now about dissemination, not just dissemination into as a publication of results, but about application into clinical practice. The rules for data handling in clinical practice are very the same as for clinical research, but they are more strict. Of course, the data has to be available, accessible, in good quality, and should be simply used for clinician. So there is also need of appropriate IT infrastructure. In the precision medicine, it's often very specific for a specific area of medicine. But there is necessary also consider more policies and also health infrastructure, facility possibilities, equipment, and also human and financial resources. And at the end is also necessary mention the security. There are more strict rules to security because of during clinical practice, the data are usually personal data. There are information about biometric data. This is not usually such deep in clinical research. And also, if we want to apply the results of clinical research to clinical practice, it is necessary to consider optimization of processes and effectiveness in clinical practice because there are requirements, for example, to user-friendly handling of information, to be there minimally user interaction, to be the system and the results very quick, accessible to clinicians. So the step is make supportive clinical decision function and integrate them to clinical infrastructures. Currently, we are working, and ELIXIR members are focusing on the field of hematological and oncological malignancies, and also onto autoimmune and neurodegenerative diseases. So at the end, how to conclude this? How can we help the field of precision medicine? How we want to help? So, during the clinical research, there is a good way to collaborate from the beginning. So bioinformaticians can help clinicians with in silico modeling, with data handling and reporting. So it means they can show them via modeling. This hypothesis seems there. This be good way. This be good way to make vet experiment on this hypothesis. This way looks like it leads to nowhere. So we can eliminate the ends with no possible results. We can also help with working on analytical study plan, and also we have tools for data management plan creation. During data collection and generation, there is good to work together on metadata definition, and also ELIXIR can help with services for ensuring data interoperability, accessibility, reliability, usability, and with the whole data management from the point of view of sharing and storage, including data security, data transmission, access restriction. It has tools for this currently available. We can help during data analysis with access to suitable computational tools and capacities to help with data quality control, and of course with application to clinical practice, so in the development of specific clinical decision support tools. But also ELIXIR itself is a broker for ELIXIR Europe tools, so he can offer you access to some tools to access and creation of specific databases needed in personalized medicine, and also with development of new necessary tools as needed from users. And also we can we hope that we will offer tools training for the tools ELIXIR can offer. So thank you for your attention, and if you have any question, please let me know.
Thank you, Lucie. And we certainly have questions, so I'll go one by one. So first was the Karel Berka, who asked, what is currently the biggest hurdle for precision medicine? If you can specify, is there data interoperability, availability, verification, or what? Just briefly tell us what is the biggest hurdle.
So the biggest hurdle. I think it's just my point of view, but there's a problem with laws, financial resources, and also anonymization process. I think the legal issues are the most hard ones, because the other ones can be handled the similar way as others data from biological research.
Okay. Robert Pergel is asking if you use the root knowledge model in Data Stewardship Wizard, or if you have your own specific customization.
It depends on what users will want. We now have no specific tool, and I think if it will be necessary, we have to do customization of Data Stewardship Wizard.
But you currently do not have any request for, let's say, specific installation. Okay. Maria Novotny, how could bioinformatics help with dealing with sensitive personal data? If you can tell us something about it.
Dealing. I think it's about security. It's about authorization and restricted access to the data, and about security transmission. I think it's the same as for others data, but it's more strict and has to be more covered.
So you think, for example, that AI authorization and authentication infrastructure can help in that matter?
Yeah, I think so.
Okay. And last question from Robert. If do you somehow use ontological methods to support semantic interoperability?
Yeah. We are working together with Czech Technical University of some tools which covered this from this point of view. In the case of marrow donor registry operations.
Okay. So Lucie, thank you for your answers, also for your presentation. To all of you, thank you for your attention. And if you will have some time, please give us a feedback and see you next Friday. Have a good day. Bye.
Thank you. Goodbye.